you all for coming. It's a real pleasure to have the opportunity to talk to you briefly about our lab's research and to have the opportunity to introduce Jamie's research as well. The common theme in all of my research is that we seek to understand how variation occurs within a species in male reproductive success. And you can imagine, you might be familiar that male reproductive success varies in pre-mating success, such as their ability to attract a mate or compete for access to mates. But what you might be less familiar with is that there's also post-mating variation in reproductive success. And that comes in that there's variation in male ability to fertilize eggs, and also in the quality and quantity of offspring that are produced, that are sired by particular males. And this topic is particularly interesting to me, this post-mating reproductive success and how males within a species vary in that success. And I study it in insects. And in insects, this is interesting because mating and fertilization are temporally separated. So females mate, but they don't fertilize their eggs right at that time. Instead, in most insect species, they store sperm. And this is, now you're looking at the inside of a female fruit fly, in case you haven't done that before. But this is her uterus. This is where the ejaculate comes in when she mates. And then sperm gets stored in these really specialized sperm storage organs, sometimes for days or weeks or even months in some species, and years in other species actually. And so because of this temporal separation between the mating and the fertilization, there's a lot of opportunities for some males to succeed and some males to fail in their reproductive success, even if they've been successful at actually mating with a female. And so the different stages where male reproductive success can be influenced is in whether or not his sperm gets stored by a female, whether or not she lays eggs or right after mating with him, and whether or not she mates with another male, which would then put sperm from multiple males into her sperm storage organs where they compete for access to fertilization of eggs. In my lab, we're addressing two questions currently. One is what factors influence post-mating reproductive success for males? And the second is how can we use this knowledge to improve pest management techniques? And I'm going to tell you briefly about two studies that we're conducting right now and how one of them led into Jamie's study. So the first study is about a group of mosquitoes in the genus Aedes. And these mosquitoes transmit organisms that cause diseases, including dengue fever. And dengue fever is, if you haven't heard of it before, it's a serious disease that can make people very sick and even be lethal. And it's spreading over many parts of the world, and there's currently no effective vaccine or treatment for this disease. So a lot of the focus on the sort of stemming the spread of this disease focuses on controlling the mosquito. One interesting aspect of this mosquito's biology is that mating fundamentally changes female behavior and physiology. So for example, when you compare a mated female to an unmated female, a female who's mated and is presented with another male is unlikely to mate with that male, whereas a female who's unmated is likely to mate with a male. Furthermore, females who have mated increase their egg production, and they have changes in their feeding behavior. And so since the 1960s and 70s, it's been known that this change in mated females occurs. And research done in the 60s and 70s showed that proteins that are transferred, so now we're inside a male reproductive tract, sorry, I should orient you. These are his testes. These are the equivalent of the prostate gland in mosquitoes. You can imagine they're very tiny. And if you take these and you inject the proteins from these glands into an unmated female, she'll go through the same changes. So that suggests that proteins from the males that are transferred during mating are inducing these behavioral and physiological changes in the female, and we call those seminal fluid proteins. And yet, these proteins were never identified. And so our lab's work is seeking to identify the proteins and try to figure out which proteins are associated with which changes in females so that we might be able to find new ways to interrupt the reproduction or the feeding behavior of these species and help to stem the spread of these disease-causing organisms. We also studied Drosophila melanogaster, the fruit fly. And similar to the mosquitoes that we studied, these females of this species go through a series of behavioral and physiological changes after mating that are also induced by proteins that are made in the male reproductive tract, so these seminal fluid proteins. But unlike the mosquitoes, a lot is known about these proteins already in Drosophila. They've been studied for many years, and particular proteins have been associated with particular changes in the females, such as we know a protein that when a female receives it, she'll be much less likely to mate, or she'll be much more likely to produce eggs, for example. And what our lab is looking at is, are males able to tailor their ejaculate, so to change the amount of proteins that they transfer to females depending on their environment? And so, for example, you can have another male present when the male's mating, and what we found is that the male will change the amount of these proteins, and that has consequences for his reproductive success in the mating with that female. In the context of studying these variation in these proteins, a collaborator contacted us to ask if we wanted to work on a project with him to study the effects of herbicides on male reproductive success. And Jamie will introduce this topic to you, so I'm not going to go further into why we're studying it. But this is Dr. Anthony Fiumera at SUNY Binghamton, and we worked together on a study of trying to look at the effects of herbicide on male reproductive success. And my side of it is to look at the seminal fluid proteins. And this study began in our lab with a former IS student, Harper Jacques, who looked at whether a particular herbicide, atrazine, affected male seminal fluid proteins. And while she was studying this, she noticed that males treated with this herbicide, atrazine, were less likely, it sort of took longer to mate than males that weren't treated with the herbicide. And that led us to this next question that Jane is going to talk to you about, which is whether those differences in males treated with atrazine from control males is due, and their mating, their sort of time until mating, is due to differences in male courtship behavior. So without further ado, I'd like to introduce you to Jamie Innis, who's been a pleasure to work with, and I'm excited to hear about her talk.
Hello, good morning and welcome. I'm so glad you guys are here. As Dr. Surratt said, my name is Jamie Innis, and I would like to talk to you about the effects of herbicides on fruit flies. And so to begin with, I kind of want to give you a couple of facts. The current world population is 6.9 billion. And it's estimated to reach about 9.3 billion by the year 2050. Considering that the average American eats about 1 ton of food per year, many people are looking for ways to increase food production. One such way is relying on chemicals such as herbicides and pesticides. Atrazine is a commonly used herbicide in the United States. It's actually the most common, and the U.S. uses approximately 76.5 billion pounds of this per year. And as you can see from this map generated in 2007, there are a lot of states that rely on atrazine. And if you look at Iowa and Illinois, Indiana, and parts of Ohio, you can see that more than 85 pounds per square mile are used. This is a lot of atrazine. And although it's effective and it targets and kills weeds, what we found from research in the 1990s is that atrazine is an endocrine disrupting chemical, or EDC. And EDCs have the ability to disrupt the production, transport, and or secretion of hormones in both invertebrate systems and vertebrate systems. And we know, for example, that vertebrate systems are affected by looking at the male African clawed frog. In 2002, Hayes and his colleagues showed that when frogs were exposed to atrazine, feminizing effects occurred. Some of these effects included larynx demasculinization as well as hermaphrodism. We also know that other organisms, such as humans, are affected by atrazine. Humans come in contact with atrazine by drinking contaminated water, handling foods that have been in contact with atrazine, or using atrazine. And in 2011, Craigan and his colleagues showed that women exposed to atrazine through drinking water tended to have high irregularity in their menstrual cycles, as well as longer follicular phases. And he compared these women from agricultural places such as Iowa and Indiana to those who weren't in an agricultural area. And there was a significant difference in the change in hormones from the women in an agricultural area where atrazine was present compared to those who weren't. And so we definitely know that atrazine affects vertebrates. What we don't know, however, is how much are invertebrates affected by this chemical? And if they are, how so? To what extent? And this is where my project kind of took off. As Dr. Surratt said, Anthony Fiumera and Harper Jacques in 2012 found that when fruit flies were exposed to atrazine, there's a decrease in mating success, as well as a decrease in the latency or time it takes for fruit flies to mate. Now, there are a lot of factors that affect mating. Some of them are visual cues, some are auditory cues, others are tactile or touch cues. And the most obvious one happens to be visual, and in this case, it's courtship behavior. And so I wanted to see what was happening between atrazine and these fruit flies. Was there a disconnect somewhere in the mating process? And the most obvious one that Dr. Surratt and I thought was courtship behavior. Maybe there's something happening with the fruit flies when they're exposed to atrazine, which leads them to have irregular and abnormal courtship behavior. And so I decided to ask the question, does atrazine affect Drosophila courtship behavior? And if it does, what parts of it are affected? And so to do this, I first had to figure out the difference between a male and a female fruit fly. And the female is on the left. She has the larger body and the black and white stripes. And the male is on the right with the darker abdomen and a smaller body. And also to answer this question, I had to figure out how fruit flies courted. So like any student, I went to YouTube and found a video. And so I want to show you a small clip of some courtship behaviors, common courtship behaviors, and I'll tell you what to look for, and then that'll be helpful when I explain how I did my actual experiment. So the male fruit fly is beginning to follow the female in this chamber. And the female has many options. She can either choose to accept the courtship behavior or reject it. And so as he continues to chase her, he also will do a wing vibration where he sticks his wing out, and this produces a vibrational song. And she can accept it or deny it, and at this point he will attempt to mount, and if he's successful, he will be able to get on top, and a successful mating is categorized as when the female's wings are spread. And so, in answering my question on whether Drosophila are affected by atrazine, I had to first expose my flies to atrazine, and we used a control group that had no exposure, a low dose group, and a high dose group. And these doses were picked on what was ecologically relevant in terms of what I would see in the actual field, a very low amount compared to the highest amount you might actually see in the environment. And then what we did was we placed a female, control female, in this small mating chamber, and I have two of them to pass around so you guys can see what they look like. But we placed a female and a male into the mating chamber, and then we began to observe their courtship behavior. So as I showed before, there was a wing vibration where they produced a song. We looked at the duration of the following that the male did, and we also looked at the number of mount attempts that the male did. And what we found was that there was no significant difference in the courtship behavior of these flies across the experimental groups. You have wing vibration on the bottom axis as well as following behavior, and on the y-axis we have the mean rate that this was happening. And if you look at the different treatment groups, there's no difference in behavior, which was kind of confusing because Dr. Surratt and I assumed that if flies were exposed to atrazine, there must be something going on, such as a disruption in their courtship behavior, to cause that latency to mate and the decrease in mating success. So we were kind of confused, and we thought, okay, maybe there's something wrong with the way we actually carried out this experiment. And so to compensate for this, we decided to do a second part. Again, we exposed our flies to different doses of atrazine, but this time we put our flies in a larger vial. And we thought that if there's a larger vial, there's more space for the female, so the male is going to have to actively pursue the female and court her. We looked at wing vibration again, the duration of following, as well as the number of mount attempts the male tried. And what we found was that there was no significant difference in courtship behavior. Wing vibration again is on the bottom, following is on the bottom, and on the y-axis we have the average rate that this is occurring. And we expected to see changes because of the larger container that they were allowed to mate in. If we also look at the number of mount attempts, which is on the x-axis, and the average number of attempts, there was no difference in the amount of attempts in the different experimental groups. And so this was confusing, but we can safely say at this point that at the doses used and under the conditions we used for this experiment, courtship is not affected by atrazine. But there must be some other mechanism that can explain Harper and Fumera's data, some other explanation that we can use to say, okay, well, there was a decrease in mating success and a decrease in, it took longer for males to reach an actual mating. So perhaps something else is going on, such as pheromone production. Pheromones are chemical signals that animals produce, and they can change the way another animal in the vicinity is responding. So fruit flies can produce a variation of pheromones that can communicate to another fly, I am willing to mate, I am not willing to mate, I am your family, or I'm not related to you. And so it would be interesting at this point for another student next year to look at the production of pheromones and to see whether or not atrazine is affecting that. And with that, I would like to acknowledge my advisor, Dr. Surratt, who is a wonderful person and great experience when I worked with her. The biology department, Ronald Tebby, who helped make the fruit fly chamber that I passed around, as well as my family and friends for helping me in this experience. Thank you.